Yep, so I think we can go ahead and get started. It's 2:05, giving everybody the 5 minutes to get started. Thank you everyone for coming to this edition of the office hours, our first one for 2025. We started this last year and we'd like to continue on with them for the remainder of 2025 as well. And just as a heads up, this is being recorded for all of those present. So we like these to be kind of a discussion and sort of just an open time to ask questions, talk about things as they are related to microscopy, to computer vision, machine learning, materials, and nuclear materials specifically, if you want to go specific in that. But we kind of have it as a chance for us to also showcase some of the things that we've been doing and working on for everybody that are our users and the community around the technologies that we've been developing over the lifetime of the company. So this time, this one, the topic is sort of what happened in 2024 and what we'd like to see happen in 2025. So that's kind of where we're at with that. If you have we have a little bit of a conversation started there. We're going to be talking about trends and things that we've seen in the past and things that we think are going to be trends in the future. So go ahead and if you want to contribute at any point in time, put it into the chat or all of you pretty much know me, so just interrupt me and because I've been caffeinated and ready to go. So just make sure you interrupt me and with your questions. So with that, we'll take a quick look back at 2024. We sort of centered this around this idea of where we have been. This is an idea because this is how we met most of everyone on this office hours. These are different places we've gone and presented our work and where we've been able to build up this community. And it also gives us a chance to kind of highlight where a lot of the features and stuff that and progress that we made in 2024 came from. So we have collaborations and work with Idaho National Lab. Obviously, we also had ones with Mivel. Many of the team here is there. The director of Mivel is with us, as always. I will also take a moment to congratulate Dr. Kevin Field for his PCASE and everything, and his Presidential Early Career Award from the DOE. So we can give him a round of applause for that effort and everything else that was recently announced. But he is also the director of MyBull. He's our vice president and chief science officer, I guess you could call him, for Theia Scientific. But then in addition to that, Anna, who is our communications and marketing specialist and is the one sort of behind the scenes directing this office hours, her and I, we went to Fermilab out in Illinois and gave a presentation there in a field and for particle accelerators. That is something that we had not been associated with before. So we've been to all these different places. I'll also mention and highlight Cleveland, Ohio, which is where we went for was it microscopy and microanalysis conference, or M&M. So that's where we had our booth down there. As you can see, we went there. Many of you also go to that conference, so we saw you there. And then we went to Argonne National Laboratory, that's here in the center. And we've got Adrian and Maria on the call here as well. They graciously allowed me to come out and piggyback onto their experiment and week-long in situ TEM experiment for there. And then I also want to highlight that we had this unique opportunity where we are an award-winning application and system. We got to go out to Amsterdam to receive the award on the big stage at Graphonicon last year. And we're looking to go back to Graphonicon this year in Seattle. So that's kind of that's sort of what the company has been able to do and what we've been doing for developing the community and growing out from there. So what we'd also like to highlight is sort of the technological improvements that we've made in 2024 and kind of go through these a little bit just to sort of highlight some of the big ones. Our tiling, which we demoed at our last office hours back in November, tracking, which we're going to see a little bit more of later today, pan and zoom, and then the reference images. All three of those were requested by the community, and we were able to turn those around and put that in there for there. And then a lot of the people on this call and a lot of our users have now had experience with the TS YOLO application, which we are looking to, and we'll discuss a little bit more later on what we are looking to do in 2025. But I want to hand this over to Kevin to sort of discuss one of the big accomplishments that we had in 2024, which is this multi-model analysis. So Kevin, if you want to talk a little bit about this, since your group has been using that feature quite extensively recently.
Yeah, sure. So, one of the things that we had a big push in 2024 for was to do multi-model analysis, or basically that means I don't know what model is going to work for my real-time experiment, so let's load up a bunch of them and run them all at the same time. And so there was a lot of refactoring in the back end of the Theia scope in the early 2024 to enable this to run on either the simple little Theoscope S's and using what we call fractionalized GPUs, or using the larger scale GPU systems that some of our users have, where they'll have multiple GPUs and just run individual models on part of that. And so that was developed as part of a SBIR project between Theia and a couple of the user groups, both at Idaho and here at Michigan, to get that integrated in. And so we have been using that here at University of Michigan over the course of the whole past year, prototyping it, giving feedback, using that. And actually, one of the most exciting things is that Justin, who's on the call, has been doing these in situ annealing experiments where we pre-implant a sample with helium that's basically insoluble in the system, and then when you heat it up, it precipitates out helium bubbles in the system, and we're trying to understand what that effect is. If you know my group as well as the group at University of Wisconsin and a couple others, there's been a lot of different models that have been developed for finding those cavities. And so when Justin was starting that experiment, he's like, what one do I use? We didn't necessarily know what one was the right one for the particular experiment because we had never done those in situ annealings. So we loaded up all the models that we had everywhere, and we're able to run those in real time with sub-second capability. And so Justin actually just submitted his first extended abstract discussing this for the ANS student conference. So he's actually going to present it as part of the student conference that's going to be at University of New Mexico here. And then we're translating that into one of the first publications on this kind of multi-model Theoscope system. So if you are interested, I'm sure Justin would be willing to share his draft abstract. We're still going through the review process, and then of course we'll keep people updated as these papers these publications start coming out. So yeah, I will hand it back to Chris, because one of the other things that we've been playing around a lot with is tracking for real time, right? So you can imagine that you want to track how many humans are crossing your front door, or how many cavities are growing in your system and what their growth rates are. And so this is something that we integrated in 2024 as well, but we're not sure if everybody knows how to access it or where that is, and the capabilities of it. So we just wanted to re-highlight and show how you can actually activate tracking. It is restricted to YOLO V8 and V11 in the current versions of Theoscope, but one of the main things that we're moving towards in 2025, and one of our kind of New Year's resolutions, is to make sure that we have it for every single model that we support on the system. So it will come in with full support for probably mask RCNN next, and then flow down from the rest after that. But we wanted to show how that is, so once it is working in all of the models, or if you have a current V8 or V11 model, you know how to activate it and run it in real time. So with that, I'll pass it back to Chris to show off a quick demo of tracking features, actually based off of some of Justin's in situ annealing work as part of it. So we're just stealing all of Justin's work, claiming it as our own, and showing it to you guys, which is exactly what managers and professors do. So we're doing our jobs.
Thank you, Kevin. Yeah, and thank you, Justin, for for this great demonstration of the tracking feature. And you can see in the background here, this is a really great screenshot of the multi-model, and then also of the tracking all in one sort of shot, that you have the side-by-side comparison that we have now available, and you can start to see the tracks for that in there. And so anybody who has a Theoscope actually has the ability to do this. And we've learned from collaborations with other users and people that of how many models you can actually run. It is quite surprising, even with just a single GPU, you can do some of these multi-model experiments and add the tracking into it. So we're here on our home dashboard, which should be familiar to most people who have turned on their Theoscope at some point. We have added some other features to that, but I've already spun up the model that has the tracking in there. The way you would do this is you could hit the play button, it'll call out this actor configuration board, and we've hidden the tracking features further down. So if we collapse some of these sections here, we've got tracking, and we have two different types of trackers available. We're going to use the bot sort today. There's also the byte track, and each one of these trackers has their own set of parameters on there that you can tune and play with. I will mention that it is key, getting these parameters correct is key to getting the tracking to work. Especially in the microscopy field, because the trackers were originally designed for tracking people. And for like live streams of videos at certain frame rates and buffering and stuff, which don't necessarily match up right out of the box with microscopy images, especially in situ TEM and stuff like that. So you do have to play around with these, and Justin and Kevin his group have figured out some of the good sort of starting points for those. So that will then spin up a model with the tracking in there. We're idle at the moment, and we're going to go ahead and go to our acquisition dashboard. And today we're going to start a screen capture and on a video from an in situ TEM experiment. Let's see if this works. Right, this is always the nervous part when it comes to real-time live demos for the system. So I clicked on the screen, I selected the window that's playing my video. Now we're doing acquisition, and I'm going to start the video here on the other side as if we were doing an experiment. I've got the tracking on, and we can start to see that we have some tracks being there. If they stay long enough, we will see them grow and go over time. And nope, there we go. We're starting to get tracks. You can start to see them. I can zoom in on them. I can pan around and look around there. That's one of the big features that we added from 2024 is this pan and zoom so that we can look at that sort of thing over time. And we saw that we can turn on and off the tracks. We can change how they look. We can change the thicknesses, the colors, all that great customization on there, but we're able to get that information in real time while we're doing the analysis, and we can see the particles move as you're doing. Oh, now that's a beautiful one right there that we're able to see the tracking from that. So that's sort of how you do it. It's a couple of clicks and off you go. And once you have that tracking and those parameters available, you can mark it as a favorite, and then it's ready to go whenever you want to. You can also download and share those parameters with your collaborators or teammates or other people who have their Theoscopes or people in the community for that. So that was a very quick demo, just highlighting the tracking, that there will be some publications out in the near future related to that. So that's some of the highlights that we had of 2024 that we did. So we wanted to say, well, what were some of your highlights? And I'm going to take a moment to breathe, drink some water, and give people some chance to jump in and mention if they wanted to. All right. Well, going from 2024, we want to go to 2025, and we have a release today that we're outputting that we'll be pushing to all the devices later this afternoon. And they include three newest features that we are really excited about having in all of this. One of them is an animation here on the left-hand side that you can see, but the first one is actually we've added Jupyter Notebook support into the system. So you have onboard notebooks that you're able to write code and Python code and copy and paste and upload and download from your Theoscope directly on the microscope. So you basically have Google Colab at the microscope that you can play with and have extreme customizability for all of your scripts and data analysis needs in there. We've also added interactivity to the vision panel so that you can click on it and it will highlight those annotations for you. This was a highly requested feature from many of you, and we'd love to get those feature requests and get them in there as much as possible. The one that we're really excited about providing for everybody is this export everything. feature, which now when you export a zip file, you can include the model JSON. I showed that we can download that. It includes the data configuration file in the YAML format so that you can upload that to other annotation software and other systems that we'll mention a little bit later on. It also includes the format in the YOLO annotations text format so that that way you can reuse those annotations directly in any of the YOLO models that are out there to use. And then also it includes the object measurements, and this is the particle measurements and everything else. This is something that was requested during the last office hours, and so we're by Kip, who is on the call and with us today. And she was an intern for us over the summer, but also has requested this feature, and we have been able to turn that around since the last office hours and provide that to the community as a way to increase our interoperability for everybody and save you as many skips and jumps as possible. So I can actually demo that a little bit right now. So here's an example of our tracking video that we had earlier. We added in the original file name because that was also requested for everybody. But we see that we have our annotations, we have all these measurements, and we want all of these things. And in the past, that was something that was a little bit wonky to kind of get out. You could use the TS-PNG package, but it was several steps to do that. Gabriella's on the call with us. She is the maintainer and user of the and super user of the TS-PNG package, and it's great that that's available, which you can use on the Jupyter Notebook support that's on board the system now. But it was a little bit of took a couple of steps. You had to export, you had to import, you had to do all these kind of things. Now we can just go ahead and say, I want to export all, and we've got the includes buttons here that you can toggle all the different information. I'm going to say I want all of this in my export, and it will start the export process. And once that is done, we will then have on there, let me see, we will have our information. It will download it in just a moment, but I've already downloaded it previously so that we don't have to wait for me to download it. But we see that we've got our CSV file with our measurements, all those aspect area, ratio, all the things that would come from Fiji or image J for you and those kinds of things. We also then have it in the TXT format. Like I was mentioning earlier, this is the YOLO format for the instant segmentation results. And then our classic now TSPNG files, but then also all the way at the bottom of our process, we've got the data config file, which has all the class names and information, and then there's our model JSON. So in your zip file, you will have what model was used, what conditions it was trained under, what conditions the inference was running, what were those tracking parameters in there, and then also what was your training and validation result configuration, all available for you to run in that system. And so that's coming out today for everybody to use and have available for all of those features. So everyone's come to office hours to here. So we would love to get those kinds of feature requests. We saw an example of us being able to turn that around. We do a release pretty much every 2 weeks. We have this iterative development process that allows us to crank through these things. So we love to get those responses and those requests so that we can put them in there for you. So what are you hoping to see in 2025? This is sort of that was a very brief taste of the very early days of 2025 and what we've been able to provide to the community. So from a more broader question and discussion, what do we see for microscopy and machine learning, and from machine learning and computer vision, and for Theia Scientific in the future? So while you're typing in your responses, I'm going to hand it over to Kevin again to talk about what we've seen as sort of trends in 2024, and then what we see are going to be the goals for 2025.
Yeah, so I'll give people just a minute, right? Because you've seen some of the new features, right? So adding tracking, including the export all. I know some of you are newer users to using models. Some of you are doing training. Some are just using models, so on and so forth. So is there a certain thing that has been bugging you the past, 2 months or 3 months doing your microscopy workflows or doing your ML? It doesn't have to be pertained to Theoscope or Theia Scientific, But we're curious about that because those are things that we want to try to challenge and tackle. So I'll give a minute just to, if you want to type it, or if anybody has something that's just like, this has been irking me for months. Why does this not exist in image J or in the Theoscope system? I'll give people a sec to voice those because we'd love to hear them. Yeah. Oh, yeah, yeah. So Justin wrote, so for people that can't see the chat, right? So Justin wrote that live tracking is great, but it's kind of just an initial first touch because there may be secondary things that people want to do, such as post-processing or even pre-processing going into the tracking. So this ties towards actually to other features requests that we have and are working towards, which is things like individual filtering based on confidence scores, right? So another request that we have that we're actually is in the next in a soon-to-be milestone would be is let's say you have dogs and cats, and you want the filter cats at 10% confidence and dogs at 50% confidence, right? Which would help with some of the tracking, as you mentioned, Justin, in there, especially with multi-features as part of that. So we are integrating that capability into it, and that part will be integrated into the tracking as well, where you can feed these pre-filtered confidences into so you can do some pre-filtering into it. One of the reasons that we added this Jupyter Notebook support is that if you had an existing confidence filter or other post tracking filter that you wanted to apply to the data, you could do that through the Jupyter until we integrate it in there. Yeah, good point. And I think we have some things on the immediate horizon that will help with filtering or post processing or pre processing as part of that. But also, what we would ask is that if there's very specific ones, like I know in high resolution TEM and some other ones, there's some very specific types of pre and post filters that may or may not be used for tracking or not. If you have those or you see those and you want them into the system, that's where you can contact us directly and we will try to get those in as quick as possible. So good point. Yeah, thanks, Justin. Is there any add custom annotations, remove annotations after the model? Yeah, so this comes to filtering. That is also a feature request. So Gabriella put in the chat, it would be nice to add custom annotations, remove annotations after the model does the detections on the images, right? So this comes towards what we have deemed as being an interactive experience, right? In a sense that you would be able to say, hey, the model got 99 of these right, but the 1% is wrong. I just want to go in and click it. That's still faster than labeling all the data myself, right? And so we are actually actively working towards that as part of some of the federally funded projects that we are working on, as well as proposed ones and for the future. So I suspect that will be coming in the near future. And that actually ties towards one of our goals for 2025, which is in the lower right-hand corner, which is make things easy or easier, right? We think it's easier than the common one, but we want to make it even easier. And so we have been working with another ML team called Supervisely that has some very nice interactive tools for labeling things, including smart annotations. If you've used it in RoboFlow, it's a similar one as part of that. We are now part of the kind of like a cohort with them trying to help co-develop theirs and integrate it into the system. And so actually Andrew, who just noticed in the chat, has been helping us as being our kind of almost beta or alpha tester of integrations into that system. I won't tell you how many annotations he has done using Supervisely, but I will say my students have to pick up the slack. Andrew's a beast with labeling that. So yeah, so the interactivity is in the pipeline as well. it takes it's going to take us a little bit more, I think. I don't know, Chris, if you wanted to comment any more on that
Yeah.
feature request.
Yeah, so the interactivity, there's you saw an example in one of the new features. Again, we have this iterative development style. So features don't come in big, they get built up over time. And so the first sort of foray into that is the clicking on the annotation in the vision panel to highlight it, that was mentioned earlier. So there's that. Supervisedly, we are working with them. We are figuring out how can we use, because their interface is fantastic. If you've used Label Me or Fiji or with a Scalable, any of these other ones out there, Supervisely has been, as Andrew said, night and day difference. It is the way to go about it, but why recreate the wheel? Let's see if we can do the same thing we did with the Jupyter Notebook, put Supervisely on board, put it at the microscope. The other part to that is this export all feature. We added in some of those sidecar files, which we are not huge fans of, but we've added in because that allows you to upload your archives directly into Supervisely so that you can then reuse those annotations directly to continue on with your annotation process and power there. So it's the start of this adaptive learning process that we have funding for and that we are looking to put in there as part of that process. So yeah, Supervisely is definitely what we're also looking at, trying to go the other direction. Instead of bringing Supervisely into the Theoscope, bring the Theoscope into Supervisely, right? There's not really any reason that we shouldn't or couldn't do that. They both run off of a similar infrastructure on the back end. So we are thinking about ways of putting that in there as one example, converting TS YOLO into a Supervisely application. They have their own ecosystem, their own app store kind of situation, similar to how we have our weight files type of thing. So we're looking at doing that as a way so that you can reuse the Supervisely if you have an onboard system to do that directly with your system. So yeah, there's a lot there that we can definitely are looking into and going from there.
Yeah. So, I think that's a great. If you have more features, feel free to do that. We also have time after this, and at the end for just open discussion, as well. And so, towards kicking off towards kind of some of those discussions, right? Some of the things that we learned in 2024 from talking to all of you guys is that it seems like everybody is moving towards more images at higher resolutions. So that means, right, like 4K, 8K images, or even larger as part of that. And then in that, that means that they have some small features, and they also have some very big features in the same image. What we're finding is that spanning that multi-scale kind of nature of the features is difficult for the models. And so, we spent actually a lot of time up until 2024 really focusing on inference, right? Really focusing on having an experience that works in real time, that takes a model that pre-exists and just runs well and runs easy. And so really, I think a lot of 2025 is actually going to be migrating towards looking at that front end of training the models and getting really good, easy to set up models for the non-ML expert, right? As doing that as part of that, and supporting the community where they're looking at 4K or 8K images and doing that part and refactoring as part of that. Or what we like to call it is the more horsepower moment. We want to have more power in our systems. And so this is actually Chris working on one of the systems that Andrew has, which is just what we call our more power system, which is bigger GPUs. If you haven't noticed, NVIDIA has announced some consumer grade GPUs that have very large VRAMs that are in price points that make sense for the general consumer, for also microscopy. So here in the very near future, we're going to be getting those through our partnership with NVIDIA and using that and testing those out and seeing if they can have better support for you guys for 4K images or higher, also high speed inference as well. So those are some of our goals, is really trying to get more horsepower, get it to the users, have an interface that works really, really well for training, really, really well for the back end, which I think we've already kind of established a good basis for that as we go through. So that's what we're driving for in 2025. But if you haven't figured it out, right, we will drive the car wherever you want us to. So if you have a feature request that isn't aligned with these goals that you think would really make a big impact to your workflow or to a colleague's workflow or the community in general, we would really appreciate that feedback as part of that. So that's kind of where we're, a retrospective into 2024 and a perspective into 2025 and where we're going to go. And so we wanted to end kind of this presentation part with just Chris talking about a new use case. So last time we showed some of the new applications of doing grain size evaluations with some of the partnerships with Columbia, who is also currently working on a manuscript that we'll see coming out here in the near future if you're curious about how that performs. And then what Chris has been working on with another collaboration is looking at this cell imaging. So I'll let Chris kind of talk about that collaboration just very briefly, and then what you're seeing on the screen here.
Yeah, thank you, Kevin. So we have here is one of the things that we've learned, especially at the M&M conference in Cleveland back in July in 2024. There was this huge push for sort of bioimaging and related to bioimage analysis. A lot of the microscopes were going into that. Cryo-EM was becoming a big thing. We went to Oak Ridge and they gave us some cryo-EM images, and they were probably the hardest images that we saw at that time. And the models started to work pretty well with those. But the thing is that in biology, they want everything, and they provide us very difficult images. And so they want not only all the cells that you maybe see on the left-hand side, but they want the nuclei, they want the strands coming out from there, and they want to differentiate every single one of these as separate instances. So we've been working with some people in the field of high content screening and drug discovery and that field. And we're looking with them to see about developing a workflow and a pipeline similar to what we have for the materials world, but have it for the cellular world. And what we have is, like here again, during our initial experiment and training a model based off of some images that they had already had annotated and available. These are freely available in the literature. So you see, this is the original image on the left, and you can see our very first model that we trained on it is doing pretty well. And in the words of the person who was working on this for us, he's like, it was shockingly good how well this model worked relative to what is sort of off the shelf available in the community and what the community is currently using. So we're really excited and really happy to see another use case and expanding out from where we've been having our sort of core collaborations from, and looking to continue to expand that stuff into 2025. Yeah, and as Kevin mentioned in the chat just a moment ago, right? The all of this is being possible because as he mentioned in sort of the perspective of going into 2025, we're moving into the model development. We originally started off like, hey, people already have models, but what we're finding is that people really love what the Theoscope does and how it works for running the models, that they want that same level of ease of use and that same process in for the training their own models. And we can see that with this new hardware coming on, with the more powerful ones, that you can train a model really quickly, and you can do it with a surprisingly low number of images. And we've really worked out that workflow and come up with this instance segmentation with the YOLO models, fixing a lot of the issues that are in the upstream packages as it's related to the instance segmentation, and then optimizing those augmentations. Really getting all that into play and all that to work has been a big thing for 2024 that we're going to look to polish up and provide as an off-the-shelf system for everybody in 2025. So that's been really great. And then I wanted to make mention, it is part of Kevin's trends and everything that we were sort of seeing, what there's what's inherent in that is this idea of big data, and the big data is not only includes what we're calling the raw images, but what Andrew, you mentioned here at the top of the hour during your introduction, that it's about the metadata too, right? It's all these other things, and that's what that exporting all is all about, is all that extra metadata, all of the stuff of, what was the tiler using, what were the parameters of that, what were those hyperparameters that were being used to train that model on, also to what day was it, what was the instrument was running, all the instrumentation parameters, like on your electron microscope, what was the beam energy, all those things. If it's an in situ, what was your beam current? All of those things need to be put in there. And also, you can then take that information and you could apply a model to that to extract information that, as lonely humans, we wouldn't be able to pull out, and find some additional insights from all that information from there. So there's this other component to sort of the data management and putting that together. And so we're trying to kind of put that in there as well. I've talked enough. So anybody else have anything they want to add?
Well, and I would just add towards the metadata, right? One of the things that we're not, now that we have a very nice established inference pipeline and kind of focusing back towards this front end, is that we are working towards integrating more and more industry formats, right? So as Andrew noted, right, in NDT, there's some very specific formats that are used for that. There's very specific formats that are used for TEM, like a digital micrograph DM3 or a DM4 file, as well as all of these other types of formats, like Zeiss has their own, and so on and so forth. And so recognizing that we need to have export our metadata, we also want to be able to integrate it where we can import those metadata that are tagged into those industry formats and then carry those throughout the rest of the inference. So we already have some support for some microscopy formats to import the images and those types of things. And so one of the other goals that we're trying to work towards in 2025 is to basically strip that metadata in the import system and then have that also coherent into the rest of the TS exported metadata as well. So you have basically end-to-end full metadata integration into the whole workflow as part of that, and doing that. So that's one of the other things is trying to aggregate what are your top priority formats that should be imported as part of the system, and have that as part of that. And in the chat, Stephen House just said, does it support the Thermo Fisher .emd format? And as Chris said, not at the moment, right? But if that's your top priority, let us know. We can look into it. It's always a challenge when you're talking about proprietary formats, because sometimes, like DM4s is well known now, but there may be a specific one that Zeiss uses that's proprietary and they don't show people what that is. There's some ways around some of those things, but we're looking into that. So yeah, not right now is HDF5 is Chris can comment towards that. So I think that's a format that we could actually integrate into it. But I don't know, Chris, if you want to talk about that.
Yeah, so the HDF5 format, we've actually had discussions to implement that throughout all of 2024, it seems like, because that is the format that's actually used in several of the models that use TensorFlow. So we are obviously a PyTorch at the moment organization or house, so everything is around that PT file. But in the TensorFlow world of model developments, that is the HDF5. And so we are looking at to maybe add support for that, only because you're also able to convert HDF5 files to PT files in certain situations and stuff. So there's some discussions about that on how we want to do that. I will create an issue after this office hours to put it onto a milestone and have that tracked, because that is definitely something we could add in. And if it's based off of the HDF5, which is also used for the big data systems and those kinds of things as well. So it's definitely something we're going to add in. It has to be added in at some point. Yeah, absolutely. There's probably a Python package that can do it. But I will I will mention as a immediate workaround, you could use a Jupyter notebook and install that Python package to read the HDF5 file for you right on your microscope with the Theoscope if you wanted to. So there is that as a workaround, which is probably why we added that in.
Yeah, so that ends kind of the presentation part, right? So we wanted to then just kind of, with the time that we have left with the next, 15 minutes, if there's more features or you're having an issue with your current Theoscope and you want us to solve it for you or give you, hey, I don't remember how to do this, or how do you do that? The floor is basically your guys's, right? So do you have any other questions or requests or thoughts that you'd like to share? Or any big experiments coming up where you might want to use the Theoscope and have Chris come as a personal chauffeur and run the Theoscope like he did at Argonne for one of the experiments.
I can do that. All right, then I'm just going to show pictures of cats.
Yeah, keep us excited. So Steven's asking, are there tutorial videos I can share with users? So I think that's a good question towards, well, and one plug for Anna as well, and a question for Anna and Chris. So I don't know if you guys want to give some updates on where we're at with that.
Yeah, so Anna, you can jump in, but I will mention that we are working on videos, making them shorter, sort of snippets of features and how to put that in there. And it's mostly been on me to just do more screen recordings. But Anna,
Yeah, we would be more than happy to produce those. Is there, okay, so it looks like, how do I use a Theoscope? Oh, okay. Yeah, and really my vision for this is to kind of fill up our YouTube channel with how-to videos and add those to the website as well. So yeah, Stephen, if there's particular ones that you want to see us do, we can absolutely do that.
so Stephen, I will mention your one question, or what model should I use? That is a question that we get asked all the time. Will this model work for my application, or which model should I use? Which we would respond with, use all of them. So you should probably get a couple different Theoscopes and cluster them together and then have them run all of the models. Which is what Kevin was actually doing with Kevin and Justin were doing with their multi-model stuff that we were talking about. Um, they have a nice system of 4 GPUs workstation system. It is the older generation GPUs. I'll mention just as a dig to get them to get the newer ones. But that so in there, in another example that we also did with the multi-model, just to discuss is that we were with Argonne and Los Alamos, and we were out there, and I think we had somewhere like 24 models running at one time. I think at that we were able to fractionalize the GPUs and slice them out, and it's surprising, like with the YOLO models especially, you're not using up that much GPU. So you can run 4 or 5 models per GPU on the Theoscope S's that are out there on there. And if you have the larger VRAM systems like the ATA 6000s that we in that one workstation earlier, you could run all the models simultaneously on a fraction each one of those GPUs with their beautiful 48 gigs of VRAM. So the idea that we're trying to solve this question or answer this question of which model should I use, both humorously but also literally just run all the models and answer that question after the fact. Because in a lot of these microscopy experiments, you only get one shot anyway. So it might be worth not worrying about, oh, we missed it because we were using the wrong model.
Yeah, and I'll note that I put a link in the chat for the YouTube channel for Theia Scientific. You can also, if you're viewing this recording and want access to it, it's just very easy to Google Theia Scientific YouTube, and we're one of the first hits that come up, or at least it's the first hit for me. I don't know what Google filters for you as part of that. But then also the website does have some very basic videos in the front end of how to use it, or just shows what that interface looks like if you have a user that's interested in using it as part of that. And then towards Chris's comments about what model to select, we actually currently are applying for funding in order to start looking at hyperparameter, on the fly hyperparameter optimization of your inference models. So when you infer, right, you may have to select what image resolution you want to pass to the ML model. You may want to select what's the confidence. There's a bunch of different actual kind of parameters that can optimize what your inference looks like. And right now, it's based on domain expertise, right? And so what we've proposed, and hopefully we'll find out in the near future if we're able to provide support towards that, is using some of these genetic algorithm type of techniques in order to rapidly in the back end, while you're doing inference, test a bunch of different parameters and then apply that. So it's not only what model do I use, but it's what hyperparameters. And this technique would basically allow you to do searches as you're inferring during real time, or do it after like on a current data set, and then it will pick, okay, this is the one that we think is the best one optimizing on certain performance factors, right? So performance factors could be, I want the fastest model speed, right? Or one is that I want the highest average confidence per image or something like this, right? And so you will have this ability to do that. So that will very rapidly rise to the top of the issues if we find or if we're able to get the support for that. Or it will stay on the milestone and be integrated after we see some of these other features that we've talked about come in as well.
Yeah.
And also, if you visited the Theia Scientific website 2 years ago, or in 2020, which is the first generation of it, I really recommend coming back to it. Anna's done a really great job at reconfiguring the whole website, putting in visuals and providing more content, and also about the different products that are in there as part of that. So I encourage everybody to jump out to that. But then I'll also let Anna plug her all of the other marketing material and things that have access to some of these little snippet videos that we make and everything else. So Anna, I don't know if you want to do that in the last couple of minutes we have here.
Yeah, just thanks everyone for joining. And you can definitely keep up with our monthly newsletter. If you're not getting that, you can either reach out to me or just email any of us, really, and we'll send you a link to subscribe to that. You can also subscribe on the website. You can follow us on LinkedIn, and then really just if you want to have a conversation with us, if it's just a quick conversation, just reach out, don't hesitate, or just send a quick thought over to us. I think anyone on this call kind of knows the collaborative spirit of the company, so please don't hesitate. The other piece, if you ever want to collaborate on some sort of content, maybe your organization does a newsletter or something like that, we would love to contribute. Reach out to us in that regard as well. So thank you.
Yeah.
Yeah.
Oh, go ahead, Chris.
I was just going to say that, yes, as many of you know, Slack, I'm on it all the time. You can reach out. We can have a quick conversation there if you're able to access Slack.
Yeah, well, that's all we had. If there's any other questions or comments, or you want to do any discussion privately, we're happy to do that immediately following this. We just really appreciate everybody coming to these office hours. We always learn something new as part of it, and that's really because you guys are coming and engaging with us. So we appreciate that. We hope that one of the feature requests we can highlight at the next office hour, that it's in there and it's working, just like we did at this one with the export all button as part of that. So yeah, thank you. And we hope to see all of you in the future. Either some of you are just down the hall from me, so it'll be very quick. But others of you maybe at microscopy and microanalysis or at your facilities as part of that. So thanks, everybody.
Yeah, you're welcome. Thank you,
Yeah,
everyone.
you're welcome. Thank you.
Have a great weekend.
Stay warm. All right.
I'm gonna end the recording.